Hello, my name is Xifeng Wei, the corresponding author of the paper Novel Recessed Electrode Geometries to Minimize Tissue Damage with Directional Selectivity in Deep Brain Stimulation. Deep brain stimulation is an FDA-approved treatment used to treat neurological disorders such as Parkinson's disease and epilepsy. Deep brain stimulation therapy is achieved by delivering charge or current through electrodes. Commonly used clinical DBS electrodes manufactured by Medtronic have 4 cylindrical contacts, generating long directional activation of brain tissue. And these are 2 models of Medtronic electrodes. And here shows one of the contacts has been turned on. It generates a volume of tissue activated long in all directions around the electrode contact. That's why we call long directional activation of brain tissue. In order to achieve directional selectivity, currently the clinical DBS electrodes are evolving from the cylindrical contacts to directionally segmented contacts called DDBS to improve current steering. And this is an example of the DDBS electrode geometry. In this paper, we call it array electrode. It has 4 columns of 16 circular contacts that generate directional activation of the brain tissue. For example, if you want to activate some brain tissue to the right of this electrode, we can just turn on the electrode contact towards that direction. However, there's some drawbacks of this DDBS electrode. High selectivity of stimulation is associated with increased current density on the surface of the smaller contact, in addition to current density being maximized at the edges due to edge effect. The high current density can lead to increased tissue damage propensity. In order to overcome this drawback, we designed some novel recessed electrode for 2 objectives. The first objective is to improve directional selectivity. The second objective is to reduce tissue damage propensity. These are achieved by designing 2 novel recessed electrodes, one with their recessed circular contact, another one with a curved contact. So the A shows their standard array electrode. The electrode contact is right on the surface of the shaft of the electrode. The recessed electrode, the contact is being recessed by distance, the distance being the same as the radius of the circular contact. However, recessing the contact by this distance may not be feasible because the contact on one end, on one side, may coincide with the contact, the recessed contact on the other side, if the electrode shaft is rather thin. In order to recess with the last step, we modified the electrode surface to be a curved surface so that the electrode don't have to recess as deep in order to achieve a uniform current density along the surface of the electrode shaft. The design motivation comes from this analogy. Imagine that the electrical current is like the flow current. Imagine a water pipe with seared ends. If one were to open the 64 holes in the pipe simultaneously, the water would sprinkle out long uniformly with increased pressure at the circumference of the holes and ends of the pipes due to the edge effect. If the holes in the pipe were recessed into the pipe, then water can only flow from the bottom of the wells in the radial direction before it diverges. Okay, so that's the rationale behind our recessed electrode in terms of reducing tissue damage and improving directional selectivity. So the rationale for reducing tissue damage comes from, recessed electrode result in decreased edge effect, which result in increased current density uniformity, which will decrease the tissue damage. Okay, The rationale for improving directional selectivity come from the recessed contact will increase the current radial projection, which will increase the selectivity. Okay, we want to increase the radial projection because if we want to project in the axial direction, we can just turn on the lax electrode contact. Okay, so this will result in increased therapeutic effects. To achieve, in order to assess the lava recessed electrode, we developed a pipeline of methods. The first step is to build lava electrode and the array electrode and recessed geometries used in SolidWorks. The second step is to use the console, which is finite element model package, to create a voltage distribution and current density along contacts. Okay, then using the current density we generated, we can evaluate tissue damage propensity using Shannon model. So on a different branch, we can use the voltage distribution to generate a Hazen matrix to calculate the largest eigenvalues to determine volume of tissue activated. And then we compare the radial projection of our lava recessed electrode as compared to the standard array electrode. So this is the results for tissue damage that come from the current density distribution. As you can see, for standard array electrode, it's highly long uniform current density distribution. The current density is higher towards the edges of the electrodes. And for recessed electrode and for recessed curvature electrode, the current density is much more uniform. It doesn't have the variability here. There's a huge variability of the highs and lows of current density for a standard array electrode. But here for this one and this one, it's much more uniform. And then we plug into the current density into the Shannon's model equation. As you can see, Shannon model separates the long damaging area from the damaging area. And for our recessed electrode, recessed flat surface electrode and recessed curvature electrode, the long damaging area is bigger. So that means the recessed electrode result in safer stimulation in terms of tissue damage propensity. And Shannon model, the modified Shannon model, the details can be found in the paper. And this is the result for selectivity. As you can see, the red shape is the VTA for the array electrode. The yellow shape is the VTA for the recessed electrode. This is when the electrode contact is at 1 volt. And the blue point cloud defines the radial direction. And the selectivity is defined by the percentage of radial volume over the total volume of tissue activated. As you can see, when it's above 1.5 volt, the percentage, the selectivity is higher for our lava recessed electrode, particularly the lava recessed curvature electrode. But at lower voltage, the selectivity, our lava electrode is not as selective necessarily. And this is because at lower current, lower voltage, as you can see, the current has to, for recessed electrode, the current has to travel some actual distance in order to reach the surface of the shaft. That will result in less radial projection. So with that, our conclusion is in threefold. The first is the standard array electrode can be modified to incorporate the recessed flat contact and curved contact designs to improve current uniformity and lower the likelihood of tissue damage. The second is our lava recessed array electrode designs, particularly with curved contact, show the increase in the radial VTA projection at higher voltages as compared to the standard array electrode. The third is the recessed electrode models have better directional selectivity and are safer than the standard array electrode models. Thank you for your attention.